Good morning, everybody. This is Don Major, Fire and Landscape Ecologist with BLM in Boise, Idaho. I just wanted to kind of welcome everybody to the Cheatgrass Die-off in the Great Basin webinar, where we're going to be talking about some of the work that's been done by USGS, EROS Lab, Stinger Graphin Technologies, and the Rocky Mountain Research Shrub Science Lab. I just wanted to kind of give everyone a little bit of background. About a year and a half ago, it kind of came to our attention this issue of cheatgrass die-off occurring in portions of Nevada and then also in Utah. So the Great Basin Restoration Initiative, along with the Great Basin Research Management Partnership and the Landscape Conservation Cooperative, came together and tried to identify a way to explore this issue of die-off kind of in an integrated fashion. So one of the things we did to start everything off was to employ the knowledge and expertise of the EROS lab in trying to help us better identify where die-off may be occurring across the portions of the Great Basin. We followed that with a more directed integrated science group where we brought together researchers to try to address aspects of causal mechanisms that may be responsible for the die-off. So what we're going to do today is kind of run through presentations by Stephen Boyte, look at the spatial extent question, and I don't have the actual, where's the title for it? Well, you'll see it in just a minute. We're going to follow that with about a 10-minute question period, and then that will conclude, and then we will move on to Susan Meyer talking about some of the causal mechanisms. So with that, I think if we can turn it over to Stephen.
And Don, if you don't mind if I interrupt for a second,
Sure.
If anyone has any questions, if any of the audience has any questions, you are welcome to type your questions into the question pane in your control panel on the top right of your screen at any point during the webinar, and I can type responses to you. But if there are questions for the presenter, then as Don said, we'll take those after the presentation.
Excellent.
Okay, so I will make Steve the presenter now.
Move that over. Okay, can you guys still hear me?
Yes.
Yes!
Okay. All right.
All right, good deal. We're ready to go. I will start over. All right, there we go. My name is Steve Boyte, and I am with Stinger Ghaffarian Technologies, a contractor to the EROS Data Center. With me here today is Bruce Wiley. He is a co-author, as is Don Major, with the Great Basin Restoration Initiative out of Boise, Idaho. We're going to talk about mapping interannual cheatgrass production and die-off using remote sensing and ecological models. So I'll go ahead and get started. This is a map of our study area. It is in the northern Nevada and southeastern Oregon, southwestern Idaho area. Our objectives over this past year have been to track cheatgrass abundance and extents spatially and temporally. We wanted to also identify cheatgrass die-off areas, and then we're going to develop a die-off probability map. Okay? The remote sensing product that we're using is eMotus NDVI. It's expedited moderate resolution imaging spectroradiometer at 250 meters. This is a 7-day composite where they take a picture of, or an image of the Earth, the conterminous US, I should say, each day for a week, and then they take the best pixel out of those 7 days, and then they composite it and make those available in weekly time steps. Now, NDVI is a normalized difference vegetation index where they take the near infrared band and subtract out the red band, and then they divide that by the near infrared band plus the red band. So that gives us a vegetation index where we can measure or use it as a proxy for vegetation dynamics, map biomass, or even use it as a proxy for net primary production. Now, the one thing about cheatgrass is its spring phenology is earlier than most other vegetation types in the Great Basin. Therefore, it produces a profile that's distinguishable from these other vegetation types. And this graph here that we're showing is showing 2 pixels, the profile of 2 pixels in close proximity. their profiles during early spring, and for 2000, 2001, and 2002. Now, the red line shows a area that is dominated by big sagebrush. It's showing its profile, and then it's being contrasted with the profile that is an area that's dominated by cheatgrass. And as you can see, there's a definite difference between these profiles in early spring. The rest of the year, they kind of follow along the same pattern, but in spring, the cheatgrass profile is significantly higher. It shows higher values than the big sagebrush does. Now, what we did was we selected a cheatgrass growing season period for spring. In this case, it was from the middle of March to the middle of May, and we used a period where cheatgrass would have senesced. In this case, it was early to late June into mid-July. And so we then created an index that contrasted the spring and summer differences, spectral differences. Now, this image that I'm showing here is a picture of the spring period image. Okay? The bright areas are areas where vegetation growth is higher, and the darker areas are where vegetation growth is lower. Now, if you look at the summer image, you can see kind of a reverse, that areas that were brighter in the spring are typically darker during the summer image, and the areas that were dark in the summer are now brighter in the summer, or dark in the spring are now dark, brighter in the summer image. And when we take and generate our index, where we take the spring image, subtract out the summer image, and divide by the spring image plus the summer image, we get our cheatgrass index. Now, again, in this cheatgrass index image, the brighter areas show areas where we think there is cheatgrass, where there should be cheatgrass growing in the spring, and the darker areas are areas where cheatgrass would not be. So we put that data into a regression tree model, and I'm going to go through this quickly because it can take a lot of time if I don't. But we have our satellite inputs, and then we have our cheatgrass index. And we use the growing season NDVI, or our spring image index, for 2001 and 2006, because what we've done is we've trained our model on Eric Peterson's data sets from 2001 and 2006. Where Peterson said their cheatgrass existed, we trained our model using 9,000 about 9,000 training points. And so we took those data sets from 2001 and 2006, and we took the spring image from those 2 years, we took the summer image from those 2 years, and we took the cheatgrass index image from those 2 years, and we added to them some site-specific variables like SERGO, or the State Soil Geographic Survey data from the available water capacity, and an elevation data set, and a wetness index, along with NRCS's major land resource area. We take these data sets, we put them through a rule-based piecewise regression model, and that generates a model output. And then we take that model output and we start adding annual growing season NDVI from 2000 to 2010, the summer image from 2000 to 2010, and the annual cheatgrass index that we generated from 2000 to 2010. We added those to those same 4 site-specific variables, and we put it into a mapping application. And this mapping application then uses all of these raster data sets to extrapolate the data from our training points to the entire study area, and we're able to generate maps for each year through our entire study area. This is the model detail. It's a strong model. The training data on 8,953 cases are R-squared equal 0.77, and the test data, the R-squared equal 0.71. So we're happy with the strength of the model. Basically what this table is showing you is how each data set is used in the development of the algorithms that were used in the regression equations, as well as how frequently each data set was used in the stratification of the rules. Now what that does is, like I said, it generates maps from 2000 through 2010, cheatgrass maps. These are cheatgrass extent and abundance maps. Areas that are black are areas where we did not estimate that there was, or the model did not estimate that there was cheatgrass production for that particular year. Now what we did when we have this time series of cheatgrass production, we can compare it to things like precipitation. And that's what this chart is doing here. This black line is our mean cheatgrass production percent through our time series. The dashed gray line is our precipitation for each year that we got from October through May. And then we have the average cheatgrass production for the entire study area for the 11 years, as well as a precipitation average for our study area. And as you can see, as cheatgrass, or as precipitation increases here, cheatgrass increases as well. When precipitation drops, cheatgrass drops. Again, in 2003, from 2002 to 2003, precipitation increased, and cheatgrass increased. And this pattern goes up until 2005. In 2005, precipitation hit a high here. In fact, it was 100 millimeters higher than the average, but cheatgrass production declined slightly, and it's a very average percent. And in 2006, precipitation declines, but it's still higher, significantly higher than the average, and we see a slight increase in the mean cover of cheatgrass in this area. Finally, in 2007, cheatgrass starts to go up, and that is also at the time when precipitation starts to drop. So what is happening here between 2005 and 2007? When we look at our maps later on, you'll see that 2005, this is when we're estimating that there is significant die-off in this area, significant die-off of cheatgrass in this area. I want to show you another chart where, again, we are comparing cheatgrass production and precipitation. This is a scatter plot. And when we look at all data points and compare all data points, there's really no relationship at all. In fact, our R-squared equals 0.01. So when we compare all years, no relationship. So when we look a little deeper, cut up the data a little bit, we find cohorts that seem to make sense and match. And this is 2000 to 2003 data when we compare the cheatgrass production to precipitation, and then we add 2008 and 2009 data points to these data points. We have an R-squared of 0.82. So we have a strong positive relationship here. However, when we look at 2005 to 2007, there's also a very strong relationship, R-squared equals 0.98. However, this is a negative relationship, was completely unexpected. We wouldn't expect that precipitation would be higher than cheatgrass production, and there would be a negative relationship, because we know that in most years, cheatgrass production is highly dependent on the amount of precipitation that it receives. 2004, represented by the circle here, and 2010 are both anomalous years. When we add those data sets to the other cohorts, they significantly drop the R-squared, so the relationship gets weaker. Now when we compare cheatgrass production with elevation, we get this chart here, and I think that there's a lot of data here, so I'm going to be short and just focus on some of the highlights. And one highlight is that at the lower elevations, cheatgrass production is at its highest. And this top line, this dashed black line, is showing 2001 data, our mean cheatgrass percent coverage for 2001. That was our most productive year for cheatgrass. The dotted black line shows cheatgrass production in 2009, which is the lowest productive year that we had for cheatgrass. And this solid black line is the mean for all 11 years in our data set. Even though this is the highest percent cheatgrass, falls into this elevation category of less than 1,137 meters. Only about 1% of the land area, as you can see from this y-axis, only about 1% actually falls in this land area. Right here between the 1,137 and 1,637 meters elevation cohorts or classes, this is where a lot of the land area falls, and this is where most of the cheatgrass is going to lie. And there's not much difference in any of these mean cheatgrass percent covered for that area. Moving on to comparing cheatgrass production and aspect, this chart shows that there's not a great difference in the mean cheatgrass percent cover, regardless of which aspect we're looking at, or even if there is no real aspect or no real slope. And so pretty much everything ranges from about 5.5 to 6.5% here. Now, when we look at cheatgrass production and percent slope, most of this area has a slope of 0 to 10%. In fact, almost 80% of the land area falls into that class. And it's also the area that has the highest percent of cheatgrass. In fact, the more steep the slope, the less percent cheatgrass that we're going to find. Now, when we do develop these cheatgrass time series productivity maps, what we can do is we can compare them and create difference maps. So what we've done is we've taken the maximum value for each pixel for all years, and we made a map, and then we compared each individual year to that maximum value map. And here I've displayed 3 of those maps. On the left-hand side, the far left, we have our 2001 difference map. This is a 2006 difference map in the middle, and then 2010 on the right. And so the areas that are consistently black in all of the maps are areas where we'd expect 0 cheatgrass percentage, or a model estimated cheatgrass 0 cheatgrass percent cover. And so when you take 0, subtract 0, you get 0. Now also in the other areas that are black, ones that are very variable, that is, for example, in 2001, these are areas where 2001 would have had the highest mean cheatgrass percent value for each pixel. That is black. When we go over to 2006, you start seeing more red areas, and these red areas indicate that, or even in this area over here, there's some fuchsia and purple colors. These are indicating that there's less cheatgrass productivity than there was in our maximum year, which is in many cases going to be 2001. And in 2010, we see even more red areas. So the productivity, cheatgrass productivity in 2010 was lower than what we see in 2006, and it's significantly lower than what we see in 2001. However, the one thing that we think we can do is we can improve on this. This is a difference map. It doesn't take into account weather effects, and we know that weather drives vegetation growth, in particular precipitation with cheatgrass. So what we're going to do is I'm going to show you where we've improved, what we think we've improved on these difference maps, so the data is more meaningful. We're doing that by developing cheatgrass production maps that have been normalized for weather, for annual weather, and then we're comparing it to our production maps that have not been normalized for annual weather. And this allows us to highlight ecosystem performance anomalies. And these performance anomalies, when they're underperforming anomalies, they are going to be areas that we think are cheatgrass die-offs. So again, another model schematic. This side of the model I've already showed you, so we're not going to worry about that. But this side of the model is where we normalize for the weather. In fact, we have PRISM weather inputs. We've taken precipitation, temperature minimums, temperature maximums, and we've aggregated those for these 5 seasons. These for each year from 2000 to 2010 are going to be put into our rule-based piecewise regression model as independent variables. We also have a site potential, which is what we're calling our long-term cheatgrass percent cover average. And this is where we took every pixel for every year and we calculated the median from our ecosystem performance, or our cheatgrass extent maps that were generated over here. So we take that pixel median and for each year, and then we calculate the pixel mean above that pixel median. That becomes another dependent variable in our regression tree model. We run that through our regression tree model. That generates our modeled or our expected ecosystem performance. So this right down here is our model that's been normalized for annual weather. We compare it to the model or the cheatgrass extents that were not normalized for annual weather, and we compare them. And we generate ecosystem performance anomalies at the 90% confidence interval level. And again, we generate 11 years of these ecosystem performance or cheatgrass die-off maps. Now this is a scattergram or scatter plot that shows the data points for all 11 years. And so we have, represented by the green diamonds, we have overperforming pixels. The red diamonds are representing underperforming pixels, and the tan diamonds are representing normal performing pixels at the 90% confidence interval. So what would cause a pixel to underperform when we normalize it for the weather? And the one thing is a disturbance will cause it to underperform. So in this case with cheatgrass, it could be something like a fire could have burned through the year before, or like we suspect with the cheatgrass die-off, is there's a pathogen that is attacking the cheatgrass and killing off the cheatgrass. And so it's causing it to underperform. Now one thing I want to, again, reiterate is that this is weather-based, that as these pixels move up and down this regression line, they're doing so based on the weather inputs. And when we look at two pixels, one that's overperforming here and one that is underperforming here, these theoretically could be the very same pixel, just from different years. So if this is pixel 1 from 2003 and this is pixel 1 from 2005, even though this pixel is shown as overperforming by our model, it has a lower expected performance score than this pixel that is underperforming 2 years later. It's got a higher actual score than this one does, but the model expects better performance out of this one than it does out of this one, and so that's why we have the underperformance here and the overperformance in this pixel and the underperformance in this pixel. And again, the anomaly threshold is going to vary with our weather conditions. So what happens when we get a year where precipitation is very high, like 2005, significantly higher than the average, but our cheatgrass percent cover is less than what we would expect? Okay, so in 2005, the cheatgrass mean was, or percent mean was 6.5%. When over the 11 years, it was 6.25%, but yet the precipitation was 100 millimeters higher than the average for 11 years. What does our map look like then? It looks like this. This is our 2005 die-off map, or our ecosystem performance anomaly map, which shows a lot of underperformance. The red indicates die-off areas. And again, this model is normalized for weather. In fact, 26% of the area our model estimates is die-off or underperformance. This made us pause because this map is much redder, shows a significantly greater amount of die-off than any of the other maps that we generated. I was reading one of Peterson's publications, Eric Peterson's publications, and he talked about in that publication where in 2005 he had field teams out in the field and they revisited 30 areas that they had visited in 2002 and or 2003, and they found in 2005 that in those 30 areas there was an 8.5% reduction in mean cover of annual grasses, where most annual grasses are cheatgrass. They found that 8.5% reduction in 2005 cheatgrass or annual grass than there was in 2002 and or 2003. So we felt better about this. We had gone back in and checked our data, made sure we didn't do something wrong, and we were comfortable that we didn't. So again, we're pointing to a 2005 map that looks like something significant was going on in regards to cheatgrass die-off. So what happens in 2006 and 2007 with those die-off maps? We have 4% die-off in 2006, 3% die-off in 2007. What it tells me is that that cheatgrass die-off is highly variable, both spatially and temporally, much like cheatgrass productivity is. One of the things we wanted to do is some validation with our die-off maps. And so the Bureau of Land Management, fortunately for us, had gone out in 2010 and they had flown areas in 2010 that they had recognized on the ground that were die-off areas. And they got up in a helicopter, they flew over this area, and they created these die-off polygons for us. We overlaid them onto our 2009 die-off map, and they matched almost 60% of the time. 60% of the time, the pixels inside the polygons were red, representing that die-off. Many of them were a dark brown as well, and that is getting very close to a die-off. Now, considering the fact that, yes, we have some modeling error, and considering the fact that, you know, when you're looking at a piece of grass, it could be as small as 4 to 6 inches high, and you're looking at that from a helicopter, you know, there's going to be most likely some error in the mapping. I think that we're very happy with our results here. The final thing that I want to talk about is a die-off probability map. Now, we have developed a die-off probability map where we took our 11-year time series of die-off maps and used, again, some attributes here that are shown in this graph or in this table here. And a quick aside here is we have temperature maximum, long-term temperature maximum. We have long-term precipitation data, and we have long-term temperature minimum data that we downloaded and processed from PRISM. The one thing that we can do is we can take and substitute these 3 data sets with future climate data from 2020, 2030, all the way up to 2100, and we can input those future climate data sets in here, and we can then project out where die-off is likely to occur under different climate scenarios. But back to this probability map, and what we have is we're happy because if you look down here at our training data, more than 9,000 training points, we have training error of 12.12%. So our accuracy here is about 88%. In this case, with our test data, our accuracy is about 86%. And when we look over here at our confusion matrix, we have user accuracies of 88 and 85% in our predicted class for our training data, and 87 and 61% for our test data. Now, the 61%, there's only 28 data points, so the low score isn't too terribly concerning. Now, our overall accuracy for our training data was high as well at 88%. So I want to move on now to the die-off probability map. The white areas are areas where we mask these areas out because they are not shrublands or grasslands. The black areas are where our model estimates that there's 0 probability of cheatgrass die-off. And then we work our way from cooler colors up to the warmer colors, going from low to high probability of cheatgrass die-off. Up here in the Oahe uplands, in these 2 areas, in the north and sections is where we see the highest probability of cheatgrass die-off, along with this section right down here just south of Interstate 80 here. Winnemucca is here, locate you, and Elko, Nevada is here, and this is US Route 95 leading north out of Winnemucca. Now I zoomed in on this map here so that we could look at those die-off polygons that the BLM generated and gave us to use. And as you can see here, those blue polygons, much of the area inside of those blue polygons are the warmer colors. So it looks like those are higher incidences or higher probability of die-off occurring. There's certainly areas outside of those polygons that show high probability of die-off occurring, but we're happy again with the amount of the warmer colors that are existing in the die-off polygons. And to finish up, I want to talk about our next steps. We're working right now on completing the development of cheatgrass production and die-off data sets for the northern Great Basin. They'll look very much like the data sets that I just showed you. We're also, as I alluded to, we're going to apply future climate data to the die-off probability model so that we can predict the probability of future die-off areas. We're also going to compare the time series of cheatgrass production maps with the monitoring and trends burn severity fire data to see if we can see any correlations between high cheatgrass areas on our production maps with fires, and then what happens after a fire occurs. How does cheatgrass come back in those areas? And then we're also finally going to compare die-off areas to topographic and edaphic data sets so we can try to pinpoint where die-offs are going to occur based on those two data sets, or two types of data sets. And that's all I have. So if you have questions now, I guess it's the time to ask them.
So again, if you have questions, please type them into your questions box and I'll field them to Steve.
Ask her if she can still hear you. Can you still hear me, Jeannie?
Mm-hmm. Yes.
All right.
Mike Pellant asks, how do you account for livestock removal of cheatgrass?
How do we account for livestock? How am I supposed to answer this? Can he hear me? Can he still hear me?
Yeah. Mm-hmm.
Okay. Okay. Well, if livestock removes cheatgrass, that could fall into a disturbance category, kind of like fire or a pathogen. If cheatgrass is grazed heavily compared to previous years, then it would show up as a disturbance, and it would be mapped as one of the underperforming areas.
This is Bruce. Also, you know that cheatgrass growing period is pretty short, so I don't know. Mike would know better than I guess, but you know, it's hard to get a lot of high-intensity grazing in a short period of time. But I guess Mike would be more knowledgeable than I am on that. But it seems like it would be hard to get very high-intensity grazing during a short period of growing when your stocking rates are usually pretty light. Anyway, or a lot of hectares per AUM.
Okay, thanks. That's the only, we have another one. Dennis Davis asks, hold on a second, I lost it. Here we go. Do you have any preliminary data to indicate the causal factors, climate, disease, other?
Of the die-off, as far as disease, I think that's what the presentation next is going to be talking about. That's Susan Meyer will be talking about a pathogen. As far as climate, we don't have, well, we're normalizing our data sets for weather, not climate. But we're still in the process of analyzing this data, and like I said, one of the future plans we have in the very near future is to compare our die-off areas with topographic and edaphic data sets. So hopefully that will give us some idea of where it's occurring. And go ahead, Bruce.
Certainly the probability of die-off map relied heavily on climate.
Yes.
Yes, so we have identified certain climatic conditions which seem to be vulnerable for cheatgrass die-offs.
Right.
And do you want to elaborate on what that was? What makes cheatgrass vulnerable?
It's really complicated model at this point.
Okay,
we can
that's fine.
that what climate variables were important, but we really can't tell you what the thresholds were. We would have to dig into these decision tree model and see what thresholds were used to separate die-offs off.
Yes.
Okay, thank you.
You bet.
Dan, I hope I pronounced your last name right. It's either Meyer or Manier, asks, an early graphic indicated statistical outliers in 2004 and 2010. I think these were the years. Could you revisit the environmental conditions, i.e. of the predictor variables during those years? Are they real outliers ecologically, or does exclusion simply make the regressions better?
Well, exclusion certainly made the regression better. What we were looking for was, no, here, let me see. I'm going to go to that, see if we can. They may not be able to see it.
Yeah, we can still see your screen.
Oh, you can? Okay, then let me go.
I mean, your presentation isn't up yet.
Yeah, let me go ahead and presentation up again. I think he's talking about this slide here. Certainly in 2010, you can see there was higher than average precipitation and significantly lower than average mean cheatgrass cover. Okay, so that's kind of an anomaly in and of itself. We would expect it with the precipitation that we're seeing, we would expect more cheatgrass production. In 2004, I think that's not nearly as surprising as 2010. Certainly 2010 really is an unusual year as well. And one of the things that, as I look at this data, I think is this another trend where we're moving into an area where things don't make sense, kind of like 2005, 2006, and 2007. I know last year in this Winnemucca area, there was a lot of precipitation and there was a lot of cheatgrass growth. However, until we get that data and we're able to normalize our cheatgrass production for the annual weather that occurred last year, we won't really know if those 2011 data makes sense or if it's underproducing or even overproducing. So yeah, the 2010 data, the predictor variables would indicate that there should have been higher cheatgrass production.
Okay, thank you. Susan Meyer asks, what would an overperforming area mean?
We're thinking on that one here.
Okay.
Yes, what would an overperforming area, theoretically speaking, could it be an area that maybe some cattle had got in there early in the season and grazed earlier in a few years, and in a particular year, maybe there was no grazing that occurred. That could generate overperformance.
Another thing is previous year's seed stock may have been very high, and so you made a lot of available seed source for a very productive year from the following year. The other thing that Steve has found is that sometimes cheatgrass will germinate in the fall and go through the winter as a basal rosette. and then in the next spring it comes on like gangbusters. So it may be that our model is not quite capturing all of that previous fall germination events and then the exponential growth that occurs in the spring related to that. So that may be a partial explanation.
Yeah.
Does that sound okay?
Yeah, that does. And the other thing that could be happening is simply the timing of precipitation may be optimal in those years where we're getting overperformance, or in those areas where we're getting overperformance. So while the totals may be similar to other years, the timing is optimal.
Okay, thank you. Dan Manier, who asked the previous question about outliers, asks, so do other variables help explain these differences, anomalies? Oh, never mind. He said you can ignore my follow-up question. Okay, we'll move on. So Brian Watts asks, many of these areas have had successful revegetation treatments within the 2001-2010 timeframe. Was this accounted for as a possible reason for change in cheatgrass?
We don't have, no, it was not.
So that would be a good validation if that's showing up as an underperforming cheatgrass. If we knew where those were, and our models are showing that as an underperforming cheatgrass, that would certainly validate our models.
And yes, if those data sets, if somebody has those data sets, that would be a nice thing for us to be able to get our hands on.
Okay, and
yeah, this is Don. Just to kind of add to that, one opportunity is that with the information that we have available that they've presented, being able to kind of step it down with some of that local field knowledge would help better inform where are areas that likely are, associated with some of the past re-veg work, whereas also outside those areas or in other locations where you have, it might be able to use local knowledge to better filter things out and then focus attention, management attention on those other areas.
okay, and Brian Watts adds, in other words, could the perceived die-off be attributed to rehab success and not a natural die-off?
Right.
Yes, it could.
Okay. Matt Germino asks, I may have missed this in the PowerPoint, but can you clarify what cheatgrass percent is? Is it percent of pixels dominated by cheatgrass? Why do values only reach 20%? Ground cover of cheat can become close to 80% or more of ground area.
First of all, we're looking at the mean overall, the overall study area. When we're looking at the mean, we're looking at the average value of every pixel in the study area that is identified as shrub or grasslands. As far as, we had some areas, and I didn't show the individual maps where you could see every piece of the map. I was just showing it as an example. I can tell you, however, that we saw in some years areas that had 87% cheatgrass cover, especially up in that northeast and north central areas. So there is significantly more cheatgrass cover than we showed in our maps.
But basically what we're trying to predict are the Peterson estimates of percent cover of cheatgrass out of Peterson, right?
Yes, that's our training set.
And so you said to me that there was not large areas that had higher than 30% cheatgrass in those Peterson maps. Is that right?
As I remember, yes, that is true. There are areas, though, in Peterson's map that do get pretty high, and that is reflected in our individual, like I said, our individual annual map cheatgrass production.
Okay, thanks. David Tart asks, does competition from other plant species play a role in die-off? Is that something for the next presentation?
Yeah, let's let Susan take a whack at that.
Okay.
What we've seen is that when the die-offs come, that's when you get some of the other buttercups and stuff coming in, but I don't think there's much that can compete effectively against cheatgrass.
Okay. Kevin Kilbride asks, this is very interesting. What on-the-ground data is known about the response of natives in areas with cheatgrass die-off? Looking for management and restoration considerations.
Well, again, Susan might be better to address this, but in the field that we were there, we were seeing Poa secunda coming back in some areas, some die-off areas. That was the one that stuck out to me as coming back.
Wasn't there an area that was devoid of vegetation in 2010, and then in 2011 there was a lot of humble mustard that came back? Yeah, I think so.
Okay.
I don't know.
Mm-hmm. Okay, thanks. David Tart asks, is a die-off a low production year or a complete absence of cheatgrass?
It can be defined as a partial or a complete stand failure. That's how we define it.
Okay. Let's.
Or if I can just interject, not to cut anything short, but kind of in time interest, is there a way we can capture these questions and be able to address them specifically back to the folks?
Well, hopefully they're listening. But yes, we have all the questions. The go-to system saves all of the questions.
Okay. I was just trying to kind of make sure we had enough time for Susan.
Oh, yeah, of course. There's just one more question. Do you want me to ask it? Okay. Sean Taylor asks, since 2010, is an outlier for precipitation versus cheatgrass. Since 2010 is an outlier for precipitation versus cheatgrass. Is it also a year of high cheatgrass die-off?
Is an outlier for, so if we're comparing cheatgrass to precipitation?
Does your 2010 die-off map have a large extent of die-off?
Oh, I hear what you're saying. It has a larger, let's see here. I don't know exactly what the percent is off the top of my head, but this is my 2010 map here. And there is, you know, there's not it's not like 2005, but there is a fairly significant amount of cheatgrass die-off, especially down over in this area here and up in this area here as well. And I think as far as die-off, 2009 might have been a little more of a, well, no, I shouldn't say that. I will leave it at what I was saying right there.
Okay. All right. Thank you very much. Don, would you like me to make Susan the presenter now?
Yes, please. That would be great.
Okay.
Susan, are you mic'd on?
I am mic'd on. Thank you very much. Now I just have to make sure I press the right buttons here. Okay, let me make my slideshow come on before I show my screen up. This is the only part that makes me nervous. Okay. And there's show my screen. Everybody can see me okay?
Yep.
Yep.
Okay. Our work started quite a bit later than the work of the folks at the Aeros lab. We only got our money at the end of the summer. So we've only been hitting the ground running for about 6 months. So if things seem a little sketchier at our end, that's one of the reasons. Our research group includes folks from four institutions, myself at the Shrub Sciences Lab in Provo, and then we have Peter Weisberg and Elizabeth Ledger at University of Nevada, Reno, and Peter is our remote sensing guy, and Elizabeth is our restoration ecologist. And then we have Brad Geary at BYU, who's a plant pathologist, Zach Onderud, who's a soil microbial ecologist, and Julie Beckstead, who's just kind of our all-purpose ecologist who also has a lot of experience with plant pathogen work. So I pulled these people together to try and we were asked to address every possibility about what might be causing these die-offs. Oops, doesn't want to advance my slides. That's interesting. Let's try an arrow. Oh, cute. Why won't my slide advance? I just want to acknowledge the fine efforts of Mike Pellant and Don Major to obtain some funding for our part of this research. You all know last year was a very tough funding year, and we were very impressed with their ability to get us some money to get started on this research, and we thank them for that. Oh, it doesn't. It's going to slow me down a little, but my next buttons don't work. Okay, just a little bit of a quick summary on what we're calling a die-off. Die-off is kind of a misnomer because it's sort of a hangover from the 80s when we had the shrub die-offs. Some of you have been around long enough to remember that, where we had stand loss of shrubs over large areas in the Great Basin, and that was really a die-off because perennial plants were dying. Whereas what's really happening with cheatgrass is stand failure, because cheatgrass dies every year, right? It sets seed and dies. It's an annual plant. So what we're really looking at is establishment or stand failure. And as was just mentioned, this is a matter of degree, but usually people don't notice it on the ground unless it's 100%. But probably it happens, would be like stand thinning. It may happen every year to some degree, but you don't really notice it unless it's 100%. And also, oops, I just lost my train of thought. My little button is worrying me here. So what are our research objectives? What we were charged with initially was to determine the causes of these die-offs, and that's obviously going to need to be understood in order to be able to manage post-die-off. And then we decided to emphasize two other aspects of the research. One was to determine what factors affect how quickly cheatgrass recovers after a die-off. And you can see from these maps that were just presented that sometimes this recovery happens very quickly, within a year. Sometimes it takes many years. So we need to understand what's controlling that. And then relevant to the questions that were asked at the end of the last presentation, do die-offs represent restoration opportunities? Can native plants grow in a die-off? And I will say that we have no evidence that whatever it is that causes die-offs has any effect at all on established native plants. They seem to be fine. It does not kill established grasses or shrubs. Whether it would kill their seedlings is a moot point. That's something we need to look at. So to try and get our brains around what the possible mechanisms, the causal mechanisms of die-offs, we made ourselves a little flow chart, and this is the way it works. Down here we have the life stages of this annual plant. It goes from a seed. That seed germinates. You get a germinant that hasn't emerged yet. Then you get an emerged seedling. Then you get an established plant, and then you get seed production on the mature plant. And the death that looks like a die-off in the spring when there should be mature plants there and there aren't, that death can happen at any of these life stages. And these life stages, this bottom panel here shows when these life stages are expected to be present. So the seeds can be present year-round because cheatgrass does form a persistent seed bank, so there can be seeds present year-round. Usually there's no growing plants present in the summer, but they could be present in autumn, winter, or spring. And then the mature plants would be present in the spring, in the late spring, and in the summer when they're producing seed. So that's just to get you some perspective on when these different variables might operate. So here's the way we set up our model. We start with macroclimate, or weather, seasonal temperature, and precipitation patterns. They interact to generate indirect effects on the plants that are these various stressors or some of them could be positive. It could be extra water, or it could be not as cold. So these maybe shouldn't have been expressed as stresses, but these are variables at the plant level that result from the interactions of these weather variables. And then they too can interact with each other, and they can affect the abiotic conditions directly, that directly influence the plants. They can also affect agents that might be involved with die-off, including pathogens and what we call predators and herbivores. And what we mean by predators there would be seed predators. So these abiotic stressors or variables are going to directly affect the plants, or they are going to affect these biotic factors, which then are going to affect the plants. And these three things can potentially interact with each other. And then as they interact, they may affect these different life stages of the plant. And depending on what the pathway is, we would have some insight into what the cause of the death or the die-off would have been. So that's a lot of fine print, but I'm going to walk you through a couple of examples of how this might work. Okay, sometimes, as we know, there are drought years, and cheatgrass simply doesn't establish. And this is not commonly referred to as a die-off because this is explained by the weather, right? The thing that caught people's attention about die-offs was that they happen independent of dry weather. If there's a drought year and there's no cheatgrass, people aren't too surprised, or if there's lower productivity. But if it's a wet year and there's hardly any cheatgrass, then that gets people's attention. But in fact, you could call this a die-off because it's stand failure. So here's the way it might work, and I mean, you could work it through any of these variables, but this particular example says, okay, we had this unusual kind of a year whereby the winter was very open, the snow cover was very low, so we had high cold stress and high freeze-thaw dynamics. The seedlings were getting shredded up by freeze-thaw in the surface horizon, and that could directly affect the seedlings such that they just die without having to be influenced by pathogens or predators. You could have a year where the cold temperature and freeze-thaw dynamics were so bad that they could just prevent seedling establishment. So that would be an example of a die-off mechanism that operated only through this abiotic pathway. Another possible die-off mechanism that's received quite a bit of attention and undoubtedly operates in some cases is herbivory. And our experience with herbivory involved the grasshopper herbivory. This was out in Skull Valley in 1999. We just happened to be doing a grasshopper study out there that year, largely because there were so many grasshoppers it destroyed some of our studies, so we decided to study grasshoppers. But we had these exclusion cages out at our study site on the White Rocks Road, and they contained the only green cheatgrass for several square miles because the grasshopper outbreak was so severe that year that they literally ate every blade of grass. There was absolutely no seed production over really quite a large area in the central part of Skull Valley. So this is an example of a die-off that happened through an herbivore action, right? But what causes grasshopper outbreaks? I don't actually know too much about this, but one of the theories is that if you have the seasonal weather and precip patterns interacting in such a way that you have very high snow cover, then you can have very high survival of grasshopper eggs, and then you can get outbreaks of grasshoppers. So what we have is an interaction of this winter weather conditions with grasshoppers to create this outbreak that then demolished all the plants over tens of square miles and caused a die-off. So that's an example of an herbivore-caused die-off. And there are other examples. There's some folks who've done a lot of work or did a few years ago with fall armyworms as a possible cause of die-offs, and that certainly hasn't been excluded as a possibility. These things have multiple causes, okay? There's no simple answer to what causes these things, but we're trying to look at all the options. The one we're the most hung up on, though, just because of our training, is disease. These are cheatgrass seeds, and they have been killed by a plant pathogen called a fusarium, which is an ascomycete facultative pathogen that's extremely common in just about every ecosystem in the world. These are very well studied organisms, very complex, but they are out there, so we're looking at whether they might have something to do with this. But one way we think that this might work with the fusariums is if you have the weather patterns that create early season water stress. Now that's not to say no precip, but supposing you have a year when there's all these little false start storms in the early fall where the seeds are sitting there imbibed, but they don't stay wet long enough to germinate, that abiotic condition at the seed level might interact with the pathogen to enable the pathogen to kill seeds in high numbers. So you'd have that effect in a year when you had this particular weather scenario. But in a year when you didn't have that weather scenario, the pathogen might still be present, but you would not get the die-off effect. Okay, the reason we think that might be going on is because a few years ago we did the classic die-off study for soil pathogens, where we got die-off soils and not die-off soils, and we put them in the greenhouse. We autoclaved half of them. We planted our cheatgrass, and the hypothesis was more cheatgrass will fail to emerge from die-off soils, and autoclaving will make this effect go away, right? Classic test of whether it's a plant pathogen. What we got was no effect. The cheatgrass plants grew perfectly well whether it was a die-off soil or not. They grew perfectly well whether we autoclaved or not. We basically had no effect on emergence from any of these treatments. So this was frustrating to us since we believed that a pathogen was involved. So we isolated a bunch of potential pathogens off of the seeds from this greenhouse experiment, which was carried out by an undergrad student of ours, Owen Boffman, who is now working with Beth as a graduate student on this project. He did a yeoman's job on this. It was a huge experiment. He basically got a null result. So we isolated a bunch of organisms off of these seeds that did not die and identified them using molecular techniques. We picked the ones that belonged to genera that are potential pathogens. We cultured them up to produce conidial inoculum. And then we inoculated cheatgrass seeds with them in the laboratory and scored germination and mortality. And we had two pairs of treatments. We either did this with dormant or non-dormant cheatgrass seeds, or we did it with or without an initial water stress period before the seeds were transferred to free water. So what we were testing here is, are dormant seeds more susceptible or are non-dormant seeds more susceptible? And we felt it should be non-dormant seeds, because if the pathogen is going to prevent emergence of a current year stand, it has to be able to kill non-dormant seeds. And we also predicted that the water stress period should give the pathogen an advantage, because our hypothesis was it will be able to grow and infect at low water potentials that prevent the germination of non-dormant seeds. So that's what we were investigating. This is what fusarium looks like when it kills cheatgrass seeds in a laboratory experiment. It forms these little tufts of mycelium on the radical end. So it is somehow sensing the radical end of the seed. It knows that's where the radical is going to come out. It is sensing some chemical signal that puts out that will cue it that seed is now susceptible to be infected. Pardon the names of these isolates. These isolates, the student who's worked on this for us, her name is Janelyn Frank, and she has an army of undergraduate research assistants. In this particular cycle of the experiments, she allowed these kids to name their own isolates. So This guy named his isolate Night Fury, which I believe is a dragon from some movie. I don't know. They had to explain this to me. But anyway, this is the pattern we got with Night Fury in this experiment. By the way, the background here, these are Night Fury's spores. It's a very prolific spore producer, so we like Night Fury a lot. But look at the pattern we got. The triangles are dormant seeds, and they essentially were not impacted by the pathogen at all. Very, very low mortality on dormant seeds, whether we did it in pure water, which is the blue, or whether we did it with the stress treatment, which is the purple. When we put the pathogen on non-dormant seeds in pure water, this particular isolate could kill about 40% of them. However, when we gave it the water stress treatment first, it slammed the cheatgrass seeds, the non-dormant cheatgrass seeds. It killed almost 100% of the non-dormant cheatgrass seeds. So this fit our model, right? Our model was dormant seeds should escape because they're not putting out the cue that says kill me. Most of the seeds should be able to escape in water because they germinate so quickly. But if we give the pathogen a head start by incubating the seeds at low water potential or under water stress, then the pathogen should be able to win and kill a lot of seeds. Okay, we didn't just do this with Night Fury. We did this with quite a few different strains. And they all show a similar pattern pretty much. It's just a matter of degree. And please, again, I apologize for these names. I did not make up these names. But some of them are very apt. Q-tip was a wuss. It couldn't kill anything. It probably isn't really even a pathogen. It killed less than 10% of the seeds, even under water stress, non-dormant seeds. Again, these pathogens were isolated out of Owen's experiment. That doesn't prove, and they are fusariums, but it does not prove they're all pathogens. This one doesn't appear to be very virulent at the very least, and we really stacked the deck in favor of the pathogen in these tests because we did the test at a temperature that the pathogen really likes. It doesn't do as well at lower temperatures. We also put a lot of spores on these seeds, so if it could do it, it ought to be able to do it in this test. So we sort of have a hierarchy of less and less wussy. We get up here, Fluffy was able to kill almost all the seeds with the water stress treatment, but it couldn't kill anything unless there was a water stress treatment. Same with Wine Spill, very lethal as long as it got a head start, but no head start, no kill. And then we had these three that could kill a fairly high percentage of seeds even without giving the pathogen a head start. So what does this mean? We don't actually know that any of these organisms are involved with the die-off yet. One thing that's interesting is that some of these isolates came from die-off soils and some of them came from control soils. So we think that these organisms are endemic in this system. They don't just suddenly magically appear when there's a die-off. What happens is something environmental, it's the perfect storm for fusarium, some set of environmental conditions occurs that permits them to go from endemic to epidemic. And whether it's particular strains that become epidemic, we don't know yet. But at least we have some candidate pathogens. These are isolated from die-off areas and they can kill cheatgrass seeds. So here's our take-homes. Dormant seeds are largely unaffected, probably because the pathogen requires a cue from a germinating seed to trigger its infection process. Most of the non-dormant seeds in free water can escape, although some strains can kill some of the seeds in free water, at least in the conditions of our experiment. If you hold the non-dormant seeds under water stress conditions that suppress germination, they can subsequently suffer very high mortality depending on the strain. So how does this relate to the field? I mean, constant water stress for 7 days is not really going to happen in the field. But we think that's a surrogate treatment that would be sort of like this intermittent wetting and drying, with long periods of partial seed hydration that would favor the pathogen that we might get in a year when there were false start storms early in the season. These soils that were in Owen's greenhouse experiment came both from the Winnemucca area, Eden Valley, and from Skull Valley, Utah. We haven't sorted out yet which strains are from which area, but basically this phenomenon occurred in both areas. They were found in both the die-off and the control field soils. Whether they could cause disease depended on the physiological status of the seeds, whether they were dormant or not, and also on the presence of environmental conditions conducive to infection. We've got a very complex genetic situation out there with different tolerances to the environmental conditions and also different levels of virulence. So we are seeing the tip of the iceberg on this. So what are we going to do to sort this out? We've already started some of this. We're going to use bait seed experiments in the field. Instead of bringing these soils into the greenhouse and planting into them, we're just going to put seeds in the field and then go back and retrieve them and see who's on them and whether they have killed the seeds. And Julie Beckstead's already working on this. She's actually working on a die-off area up in Washington that's a little bit closer to her. She's in Spokane, but it's already clear that we've got a lot of the same types of organisms that are operating up there. So that's how we're going to obtain more pathogens, and we're going to do that also in Skull Valley and Winnemucca this spring. We're going to identify and screen additional candidate pathogens from those seed bait experiments and also from seed bank samples, or if we get lucky and have a die-off and can actually find dying seeds and seedlings, we'll isolate off of those. We'll take a harder look at the environmental component of the disease triangle. You all know the disease triangle, and involves the pathogen, the host, but a very important component is the environment. And for instance, we have manipulated water potential a little bit, but we want to manipulate temperature. We want to do hydration, dehydration cycles, different environmental conditions to see if that will clarify how this might actually operate in the field. And then to move to the field in a manipulative way, we need to develop durable dry inoculum. So far what we do is we grow up the inoculum on Petri dishes. We rinse the Petri dishes with sterile water, and we get the spores in a suspension of sterile water. And then we control the concentration of spores in the sterile water, and we pour those over the seeds in the Petri dishes. But that's not a very practical way to do a field experiment, so we're working on ways to get dry inoculum, and probably this night fury that produces this wonderful abundance of these what we call banana spores. These are the durable spores of Fusarium. It'll be the first one we work on to try to develop this dry inoculum. And once we have dry inoculum, then we can carry out manipulative experiments to elucidate these mechanisms by trying to create mini die-offs with inoculated seeds. It's like we inoculate the seeds, we put them in the field, and then we manipulate water. We do it at different seasons to manipulate temperature and see if we can create mini die-offs in our experimental plots. We also, and this is Zach Onderud's, one of his main pieces, is to figure out how to quantify how much of these different pathogens are present in the field and in the bait seeds. Because we've got a whole raft of candidates, but just isolating them off Owen's seeds gave us no indication of their abundance. But there are really cool molecular tools for quantifying how much of different pathogens there are in field, in environmental samples, basically. And then lastly, we're going to, assuming we get some good die-offs this year, we don't get to control that, unfortunately, we will be planting cheatgrass into die-off and control soils, following seed and seedling fate, and doing some manipulations like fungicide treatments with different classes of fungicides to try to figure out exactly who the culprit might be. So this is a lot of work we have planned for the next year or two. I'm going to talk about Peter's stuff. Peter Weisberg's our landscape ecologist working on this project. I got these slides from him, and he kind of jumped in the deep end, and he didn't paddle around in the shallow end very much. So I just want to tell you why do we have a remote sensing component, and how is it different from what the EROS people are doing? Okay, their approach is to say how much cheatgrass cover should there be on this year based on the weather, and then if it's less than that, maybe that's a die-off, or that's caused by disturbance, and one of the disturbance variables is a die-off. Whereas Peter's approach is just going to be to compare across years in a given spot. So is there less cheatgrass or more cheatgrass in a particular spot this year than last year? Because what my fantasy, the reason I wanted to have this remote sensing thing is my fantasy is this wonderful animated sequence of maps that show how the dynamics of die-offs in space and time across years, and in a small geographic area, not this very large-scale stuff the EROS people are doing. What we want Peter to do is say, okay, here's a spot. What's the die-off history of this spot? When was it a die-off, and when was it not a die-off? And after it became a die-off, how long did it stay one? How long did it take for cheatgrass to come back? So the idea is to get up close and personal with these die-offs rather than back off and look at these average indices and stuff, which that's obviously very powerful, and these people have come a long way. I compliment them. Even the difference between that talk and the one at the range meetings last month was impressive. But I think there's still a place for this, using the remote sensing approach to try and address causality more directly, and that's what Peter's going to try to do. He's basically using a similar approach on the image analysis, which is this NDVI approach that's already been explained pretty well, so I won't dwell on that. But we're going to work in the Winnemucca area and also in the Skull Valley area, and we're going to use these differences between years to quantify the changes we see as die-off, new invasion, just productivity increase, burn, whatever it might have been. So again, we're just getting started on this. The first thing we did is we went out last fall while the cheatgrass was still present as standing litter and attempted to estimate the previous spring's cover based on the fall standing litter. So already we have a disconnect because that may not be very accurate. But we had to get started on something, so in both the Skull Valley area and the Winnemucca area, we went out and collected this training data for Peter's model. And he's gotten it to work fairly well for Winnemucca. He still hasn't gotten it to work too well for Skull Valley. Also, the other problem he's having big time is that using fall 2011 data as your training data means you're trying to use cover data from the satellite imagery from spring 2011, and we all know what spring 2011 was like. It was probably the cloudiest spring in the last 30 years. So far he is struggling with cloud occlusion. So in his maps, all this gray stuff is stuff that he just cannot see. And it turned out that a lot of his training points were actually in places that were cloud occluded because we couldn't get the cart and the horse going in the same order very well to start with. But he's definitely got to start on this. And again, this is just to show 2010, few clouds, 2011, amazing cloud cover. And his criterion was he had to have 3 pixels that he could really see during the period of maximum spring growth of cheatgrass before he was willing to say it wasn't cloud covered. So he used a very strict criterion. They're thinking they could relax that and maybe improve this a little bit. But the red areas are, we just made an arbitrary elevation threshold of 1,700 meters that we're not considering. It's not really cheatgrass country, and we just arbitrarily said we're only considering what's below 1,700 meters. So the red is stuff that's been masked out. And the gray is stuff that was so cloud covered he couldn't see it. So you can see we're not there yet on this. But I mean, he's only had the training data for 2 months. So we've got a long ways to go on this. The training data, this is the predicted cover from the training data. This is the field, this is the field measured cover. This is the predicted cover from his model. And it's not as pretty as Bruce's, but it's pretty good. R-square of 0.53. And so our plan is to collect a lot more training data this spring, and hopefully it won't be so cloudy, and do a better job of training Peter's model. He did get some results, though. He was able to show that there was a lot more cheatgrass out there in the areas that weren't cloud covered in 2011 than there was in 2010. 2011 was a very wet year. It's not in the other data set yet, but if they put it in, they might see some stuff. So he could detect differences in cheatgrass cover across years, which he then interpreted in terms of change. And what he didn't get very much of between 2010 and 2011 was die-off. If anything, we got increase, and again, this is confounded with the weather. As mentioned earlier, we got an increase because it was a more productive year. There are some areas that look like die-offs, but he has told me that many of these areas are artifactual. They're not really die-offs, because he's still working on figuring out how to interpret these images. He did find one that looked like it might really be a die-off. It just wasn't a die-off year, for one. So he zoomed in on that, and to make a long story short, it might be a die-off, but based on the Google imagery, it looks more like a burn, and he has not followed up on this yet. This could be a very powerful way to map burns as well, because I know there aren't good burn maps. And Peter's intention is to take, once he gets the technique down, is to take this all the way back through the Landsat imagery, to the 30 years of Landsat imagery, and do year to year comparisons to track the appearance and the persistence of die-offs through time, and get me that neat movie that I want to watch. But at least in the Winnemucca area, in the area that he could look at that wasn't cloud occluded, he didn't find anything that looked like a real die-off. So again, this is his problems, the clouds, but we may not need to have every pixel, so he thinks maybe we will switch to some MODIS imagery to combine as well to make our maps more complete. We need to do a lot more validation in Skull Valley, or excuse me, training data collection in Skull Valley, because the model didn't fit very well in Skull Valley. Could be due to the inexperience of the people I had collecting the cover data. Maybe the cover data wasn't collected in the same way that it was collected in Winnemucca. So we need to work on that, get people, get some group training, make sure we're all doing this the same way. But by summer, we should have a better procedure. The good news is the NDVI seems to work okay. If you get good training data, you should be able to convert that to cover values pretty well. And he did this compositing approach, taking the maximum value across months within a season, and that worked. So he's pretty happy since he's only been working on this for 6 weeks. So here's his laundry list of everything we have left to do, and his little note on his PowerPoint was a lot of steps. So we're not even close on this yet. He does have a student to work on it. We are short on money on this project. Well, we have some, which we're grateful for, but we definitely don't have what we thought we needed. So he does not have a dedicated student to do this remote sensing analysis. He's doing a lot of it himself. The student we have at Reno, Owen Baughman, will be working with Beth on these restoration experiments. This is the more of the field aspect. So Peter is making progress. So again, the reason we want Peter's stuff is so that we can follow these die-offs through time on a fairly local scale. I've been working out in Skull Valley for over 20 years, and we've had die-offs before, And I sort of remember them, and they didn't really penetrate my consciousness as not drought-related for the first few years. There are no records. I can't remember where, when, what died off where. Even in our exclosure, we have had die-offs in our study exclosure. It's a big exclosure. It's many hectares. And there have been die-offs in there, but we don't have any records of that. So it's very hard to get your brain around what the process is. So hopefully Peter is going to be able to tell me what is the history of die-offs in the White Rocks exclosure. When did they happen? How long did they persist? When they went away, what was the process? Because the process by which they recover is as important as when they occur, particularly from the point of view of soil erosion or restoration or any of a lot of things, or the invasion of secondary weeds, you name it. We need to know what's controlling how long these things persist. This is one of Owen's study sites. It was a new die-off in May based on his seed bank data, May 2008. May 2009, it was still there, pretty strong. By 2010, it had filled back in with cheatgrass. Why didn't it do that in 2009? That's the kind of questions we're trying to figure out. And it went straight to cheatgrass without an intermediate annual dicot phase. So here's our little picture that we like to make these schematics to sort of focus our attention on the things we need to look at, either in the field or as in our causal mechanism experiments. So here we have the stand failure. So one of two things happens. Either it reestablishes from the carryover seed bank, and you're right back where you started. And in that grasshopper die-off I showed you, where the grasshoppers ate everything over tens of square miles and there wasn't a blade of grass except in our exclusion cages, what do you think was there the next year? The next year, it looked like nothing had ever happened. It was a solid, full stand of cheatgrass. So it reestablished from the seed bank. That's common. That commonly happens. We had another one in Rush Valley where we observed the same thing. One year cheatgrass was gone, the next year, bingo, it was back again. But sometimes it doesn't do that. Sometimes there's a stand failure, and then it fails to establish from the seed bank. And there are some possible causes for that. It could be that whatever caused the die-off, if it's a pathogen, is still operating and is killing the second year seedlings too. Or it could be that there's no carryover seed bank. I'm not talking about black fingers of death today. I didn't think I would have time to go into that component of things, but this is another pathogen that's out there in large quantities that also kills cheatgrass seeds, but it kills dormant seeds. So it kills the seeds that don't germinate and emerge. And if it's very abundant, it can take out all the carryover seed bank, in which case that would be a reason why cheatgrass would fail to establish from a carryover seed bank, because there isn't one, because BFOD killed all those ungerminated seeds. It's usually not that extreme, but it could be potentially. And lastly, if it stays a die-off for more than a couple of years, if you have bad recruitment years, say there's a drought the next year, then the site can deteriorate to the point, mainly by the loss of litter, that cheatgrass is going to have a tough time recruiting back in. So we think there's two scenarios for this persistent die-off, where it doesn't reestablish from the seed bank the next year. If there's an annual dicot seed bank, this is those tumble mustards and burr buttercup and annual kochia and tumbleweed, that is Russian thistle, halogeton, a cast of characters who vary from innocuous to awful, right? But those are the annual dicots. They recruit from the seed bank and dominate the site. They permit the litter to build up again, and then cheatgrass can reinvade. We know it moves around readily. It disperses readily. So it can get back in there even if there's no seed bank, if the site is favorable. But suppose there's no annual dicot seed bank, then the thing is just going to sit there and have no plants on it for a couple of years. It's going to lose all the litter and be very unfriendly for the establishment of cheatgrass. It's going to take time for the annual dicots to disperse in build the litter up again so that cheatgrass can reinvade. So why can the annuals
Did we lose you, Susan?
Can you still hear me?
Okay, now I can hear you.
I'm good. I'm good again?
Yes.
Yep.
I just had a little note that said I didn't have audio for a second. So we think the annual dicots, and this has been known for a zillion years from early studies up in Idaho from 40, 50 years ago, that there's a severe disturbance. The annual dicots come in first, the weedy annual dicots, and then cheatgrass can reinvade. So here's some pictures of what that might look like. This is a die-off that Owen studied, and he knew from the seed bank characteristics that this was the first year of the die-off, and you can see there was very thick litter from the previous year, and that litter was full of seeds, and the next year it was business as usual. This die-off did not persist. It was a transient die-off. That's one kind. Then you can have this kind. This is a die-off that, based on the seed bank characteristics, he determined was more than 1 year old. It had many fewer seeds in the seed bank, and you can see the litter's already starting to go. It's going patchy. We came back to this die-off 2 years later, and it was still a die-off. The litter was almost completely lost, and annual dicots presumably had recruited in. They probably weren't there as a seed bank. They probably had to recruit in, and I mean, these things are very good at recruiting in. I mean, isn't tumbleweed the classic disperser, or tumble mustard? Why do you think they tumble like that? They go looking for severe disturbances to recruit into. Another thing that makes a die-off a really happy place for these dicots is that they're nitrogen enriched, because when cheatgrass fails to grow, it leaves a pool of available nitrate in the soil, and that makes these annual dicots happy as little clams. So they go looking for these nitrogen-enriched, cheatgrass-free areas to recruit into, and they do recruit into them. So this is 2010, and I can tell you, I'm sorry I don't have a picture, but the cheatgrass is now, in 2011, it had started to move back in to this area. So this die-off will be it will have taken it 5 years to recover. Now, why it didn't recover the first year, we're not sure. But once that litter starts to go, then that changes the whole dynamic of the successional trajectory. Okay, we don't have any data on this yet, so I only have one slide. So the question is, if there's a die-off, that means cheatgrass just controlled itself, right? So you have cheatgrass control. So does that mean you can seed? Is that a restoration opportunity? And it depends on several things, right? If native species can establish from seed under the conditions that prevail after the die-off has occurred. If there's a cheatgrass seed bank present and it just recruits back, it's a transient die-off, you probably will have thrown your seed away. If the pathogen is still operating and if it can attack the natives, that's not going to work. If the site has deteriorated so much in its physical conditions because of loss of litter that the natives are going to have a really hostile environment, that could also prevent your native seeding from taking. So it's not immediately apparent that die-offs are going to be restoration opportunities, but we want to investigate this in detail because this naturally occurring control happens. We want to know whether it presents an opportunity. So the way we're going to approach this, obviously first we're going to find out if these natives we want to seed are susceptible to these causal agents, especially the pathogens, and that's going to be fairly easy to do. And chances are fairly high that yes, native grasses are susceptible to these fusariums. There's no reason to think these fusariums are host specific, but we don't know that yet. And then, assuming we get some good die-offs, we're going to do these precision seeding experiments where we plant these natives into die-offs and controls with and without controlling cheatgrass some other way, use treatments designed to protect the seeds from different die-off agents, use litter use mulching techniques to ameliorate the seedbed conditions. In other words, manipulate everything we think might have to do with it. And then these are going to be these toothpick experiments that Beth has pioneered so that we can track individual seeds and seedling fate. And this also means that if the seed doesn't come up, we can find out what killed it. And we can find out which pathogen killed it using Zach's molecular techniques. And lastly, if we get to the stage where we're going to do these mini die-off experiments, we will put native seeds in the mini die-off experiments as well. So in summary, we have a lot of different approaches we could take, and it's going to depend a lot on what kind of a die-off year we get. We can't afford to sit around for 2 or 3 years waiting for that place in the graph that's the magic perfect storm year for high die-off to study this phenomenon. We've got to approach it from as many angles as we can. So that's where we're at with this. And this is where we hope it will all go. You should have seen the picture of Owen after he did this. He has a very large red afro of curly hair, and it was completely covered with cheatgrass seeds, and he had a very grumpy expression on his face. But he has not given me that slide to share with you. Okay, let me see how I give myself back to you.
Well, if you have questions, you can leave it on just in case people want to refer back.
Okay. I think we still have some time.
Yeah. All right, so Robert Burton asks, once you isolate the pathogens with the most potential, are you considering how sensitive these may be to soil temperature extremes, i.e. fire events, fire behavior? And if so, how long the effects may last?
That's a good question, and we look at this disease triangle thing. We will take that into account. We've done those kind of experiments with our other little critter, the black fingers of death. We've looked at how well it survives burning, and it has very high, it actually survives burning better than cheatgrass seeds do.
Wow.
Yeah, that's a good question. It'd be nice to know if there's a way to take it out, for one thing, because, you know, you see something that can kill cheatgrass this well. If you're kind of a biocontrol nut, you think, gee, maybe we could use this for biocontrol. I'm not even going there yet, but we would definitely need to understand how it persists in the field. And we will be doing those kinds of studies, assuming we can get enough funding. That's a whole other layer of thing to do, to look at the fire side of things. But yes, we are interested in that.
Great, thanks. Dirk Netz asks, how many of the fusarium isolates cause mortality over 50%? Have there been any experiments conducted to see response created by these isolates and the presence of other organisms that occur in the rhizosphere?
That is also a very good question. We are just barely getting started on this, okay? We've tested maybe a dozen, well, let's see, maybe 15 isolates, and probably half of them can cause mortality with the water stress treatment of over 50%. But this is a really artificial situation, right? We're at the absolute baby steps of figuring this out. Julie had a student, an undergrad a few years ago, who actually put combinations of different organisms off of cheatgrass seeds in experiments together to see how they would affect each other, to see if they would compete. And he did get some interesting stuff. He had a fusarium in his study, which interestingly did not cause any mortality on his non-dormant or dormant cheatgrass seeds in water, but they did interfere with black fingers. When he put the fusarium and the black fingers together on seeds, black fingers wasn't able to kill as many seeds. The fusarium didn't kill any anyway, so it wasn't a fair test for the fusarium. But yes, that's a good question, and there's the whole, and this is where Zach can help us too, because the soil microbial community is vast and complex, and to think you can reduce it to a half a dozen pathogens is pretty simplistic. We're just hoping we get lucky and that there's one or two major players out there. But yes, we're going to be, his technique he can actually take environmental samples and find out everybody who's in them and how much of each one. So we have some very powerful tools to look at that. Thanks.
Right. Oh, were you going to say more?
No, that's good.
Okay. Charlene Singh asks, how do you measure freeze-thaw dynamics? What attributes do you feed into the model for this?
That is a good question, and I wish Zach Andrew was here to answer it. He and Jeremy James up at the Burns ARS unit have been doing work on this with natives, actually, looking at they did this very elaborate experiment where they controlled the depth of the snow in artificial manipulations, and then they had the things censored up out the wazoo, and then they had seeds and they looked at seed fate. But I don't know the details of how they measured all that. We'd have to talk to Zach, and I don't think he's on today. But that's a good question. It's not simple.
Okay, thank you. Don Major asks, if folks in the field think they have found a die-off, what info should they collect?
That would be very helpful to know. They should, if they think they found a die-off, it would be helpful to know whether this was the first year it was observed. Is it a new one? How much litter is there? How big is it? Are there any remnant perennials? Are there any annual dicots present? What would the annual dicots be that are present? A good location so we can go have a look at it or find it on Peter's maps. That's what comes to my mind, but Don, I think you and I need to sit down and be more systematic about that, because it would be neat if we could just come up with a data sheet for people to fill out. We could put it on some website somewhere and say, help us figure out die-offs. If you find one you think is a die-off, fill out this form and send it to somebody. I think that would be really helpful, because like I say, it is impossible for even the human brain, the marvelous computer that it is, to keep track of all this stuff in your head. You really need people out there helping you find things.
Yeah, this is Don, just to kind of add to that a little bit. Hopefully before spring comes on us, working with Matt Germino and USGS through the ReNet, I believe it is, we're hoping to maybe have at least a location where folks could, once we kind of flesh out what is the kind of the bare bones information we need, have a place that could be provided.
That would be great.
Awesome.
Owen Bauman also added, if you think you find a die-off, take some soil seed bank samples in the die-off and in the closest cheatgrass monoculture, then compare seed densities.
But don't send them to Owen to compare seed densities. Owen did it. We have a whole other data set on the seed bank dynamics on these die-offs that I didn't even mention today, that's soon going to be going to press, so that information will become more available. But the whole black fingers interaction thing is very interesting, and you can tell from the seed bank samples how long it's been since the die-off occurred if you have a reference area that you're comparing it with. So yeah, we need to teach people how to at least take these samples and what the minimum number would be that would be useful.
Yeah.
Great, thanks. Well, that was the last question, so you can take it from here, Don.
All right. Well, I just wanted to thank everyone for attending, and special thanks to the presenters and facilitator. I just want to say thank you very much. And if you have any questions, feel free to direct them at the presenters, as well as send me an email. And I think with that, we will say goodbye.
Okay.
Thanks, Owen.
Yeah, thank you very much.
All right, thanks everybody. I'm going to close out now.
Awesome. Can we get can you send me the stuff on the presentations so we can get that out?
Will do. Oh.
Thank you.
Okay.
Just the recorded stuff.
Yeah. Okay. Bye.
Awesome. Bye.
Do you want these PowerPoints, Don?
I think it would be awesome if we could archive them in a location that folks that we want to go see could go.
I try to make them so they have enough text they make some kind of sense, right?
Well, you're
But not so much
on.
that people can't stand it. It's very hard to draw the happy medium there.
Okay, thanks. Talk to you soon.
Yeah,
All right.
bye.
Thanks, everybody. Great job.